Okay, so today I kind of wanted to go through lab number 3 electronically, give you guys some tips and supplement everything. Here are the pre-lab questions. You can take a look at those. We'll choose 2 of those for Friday, and we'll go through each of these 4 parts. For the most part, the 2 main concepts are going to be solubility, and that's how something is able to dissolve into water. Is it able to be disassociated by water? And the properties of water that make that possible, we're going to go over those. And then we're going to talk about pH, which is essentially a measure of how acidic or basic something is. More acidic compounds more acidic solutions are those that have a lot of those hydrogen ions in them. So essentially just a proton because it's H+, and there's no electron there. So the more of those you have, the greater acidity you have, and the lower the pH. Okay, so this is the cleanup stuff. So today we're going to be working with something called HCl, and this is an acid. This is a strong acid. It's not enough to really hurt you, but we'll definitely take some precautions when we're using it. The reason it's an acid is because it donates that hydrogen right here. It donates it right into the solution because the chlorine has a negative charge and the hydrogen has a positive charge, but they would rather associate separately. So the minute that they go into a solution, they separate into ions, and that's why it donates this hydrogen, thus adding to the acidity, lowering the pH. Okay, so here are the basic concepts for today. Essentially, we're going to see how molecules react when they're dissolved in water, and if you can dissolve 2 molecules at once, things like that. And one of the main things we're going to focus on is pH and how that affects things. We're also going to take a look at hydrophilic and hydrophobic molecules. Can they dissolve the same way? We're going to see some of those clear differences right in the lab. So you can watch this little video right here. This kind of goes into how the structure of oxygen, since it's polar, these bonds are not being shared equally, and that gives the oxygen, it's pulling stronger on those electrons. It gives it that small negative charge, and since the electrons of the hydrogens are being pulled, they get this small positive charge. And what that does is it allows them to not only bond with each other, and we can watch this video.
We offer 50% off your package of choice this tax season. Your refund is waiting.
This video shows how many drops of water can go on a penny. The reason the water doesn't just fly off is because all of those oxygen and hydrogens are bonding with each other with those weak bonds. And this is one of the strengths of water. The same way that these can bond can lead to interactions with other solutions. So this is solubility. This is essentially that, see right here, you can take this salt crystal, which is made of sodium and chlorines, and essentially the water perfectly shields it with these positive hydrogens. Remember how they have that small positive charge, and they go all around this negative chlorine. And the negative oxygens, with the slight negative charges here, they go around the sodium. And essentially, a lot of compounds can be broken up by water like this, and this is a highly soluble compound. When you put salt into water, it disassociates. Sucrose, on the other hand, though, is this solid, covalently bonded molecule. It's not going to disassociate the same way as salt crystal will. So we're going to see those differences in the lab. So the other big thing is that we're dealing with acid today. We're going to see how that changes the solubility of a solution and how we can stop acidic or basic solutions from changing the pH when we add a buffer. So acids, for the most part, let's say go back up to the HCl, right? This is an acid because it's going to donate this hydrogen. When you put more of those hydrogen ions into solution, it becomes more acidic. Whoops. So the way you resist that is with something called a buffer. So H2CO3 carbonic acid is something that's found in your blood. and your blood maintains a pH of 7, which is the neutral pH. It's an equal amount of base and acid, and this is where physiologically your cells and everything in your body needs to be operating at. Proteins in which there's too much acid or too many bases, they won't operate correctly. So what a buffer like carbonic acid does is it disassociates into a base which can accept these H plus ions, but it also donates its own H plus ion. See, it's not an acid though, because it's an acid only if it's donating just the hydrogen ion. But in this case, since it's donating both, either bases can react with the hydrogen and become water, usually an OH plus the H can become water, and then this H acceptor, this base, can accept extra hydrogens, these extra hydrogen ions that come in a solution from the acid. So if we pour HCl in, your body has a base that would accept all of those hydrogen ions. So you can definitely watch this video and it can show you how they kind of connect together. So the buffer we're going to be using is based off glutamic acid, which is just an amino acid. So here's a generic amino acid in solution with all those waters, all the small negative and positive charges in this aqueous solution. This carboxyl group actually loses its hydrogen and it moves over here to the amino group. And now you have kind of this plus-minus molecule. So that means that this rearrangement didn't change the amount of hydrogens, or protons in this case, because they're just little H+. Because hydrogen went from here to here. It never was donated to the solution. So let's go to MSG. So here's glutamic acid. And so you can see right here, there's an OH group, and here there's an OH group. So under the normal circumstances, this OH is going to donate here, but this OH is going to be donated to solution. That's why it's an acid, is because it's given up one net hydrogen ion into the solution. So we can change this into a buffer, and that's what monosodium glutamate is. We're not going to change that. We just have monosodium glutamate, and this is what is labeled as MSG. The deal here is as though this hydrogen in aqueous solution will go here and bind, but over here we've replaced that hydrogen ion just with a sodium positive ion. So that's just going to disassociate, and the sodium's not going to change the acidity. And so now we have kind of a neutral buffer. It can either bind with other bases that it encounters right here. So say like a hydroxide ion, it could form water. And then right here it can accept another hydrogen ion. And that can kind of take up that acid that's being put in, those little H pluses. So we'll go through this. This will all be done in lab. And we're going to put in MSG with HCl. And we're going to do it with water too, and you're going to test the difference in pH. You're going to see if MSG acts as a buffer and keeps the pH constant by kind of gobbling up all of those extra hydrogen ions that HCl is going to donate. Okay. Next thing we're going to do is we're going to see if, once we've saturated something with MSG, can sucrose also dissolve in there? Can two things be soluble at once, or are all the water molecules taken up? We're going to use a control where we just have H2O and sucrose, not both. So we'll see if both can be happen. Okay, the next thing we're going to do is we're going to see if fats and lipids can dissolve in water, and whether they can dissolve in oil, which is a lipid itself. And we're going to see right away that those hydrophobic molecules are going to have a harder time. Okay. One example of pH that I want to show you too is, say that we are in a cell right here. The pH in here is about 7.4. Remember, 7 is around the pH, so that's the concentration of hydrogen ions where things are pretty normal for your body. There are organelles called lysosomes, though. These are responsible for digesting things. That can be things in the cell that it's recycling into parts like amino acids and carbs, or in the case of white blood cells that actually eat bacteria. They like get bacteria and they put it in here and fuse it, and it digests because this pH is 5.4. So that's more acidic. That means there's more hydrogen ions in this area. and there's 100 times more actually, because it's on a logarithmic kind of exponential scale, and they digest things. So that's your physiological example. I also put up a video on how cancer cells take advantage of pH.